Oh, good afternoon, microbiology 221s from Camden County College. Today is day 4, Saturday for me. And I'm going to start with a little bit of sort of housekeeping, what we need to do to keep our lab space clear. I've given myself some new isopropanol. You might remember last night I took the inoculating loops and disposed of them, and I do have a new cup of disinfectant bleach, what was left, what was remaining, and it's a day to make new disinfectant. So I'm going to go through that with you again. I'm going to move my stain set back sort of out of the view here, and I'm going to really quickly make another 10% bleach solution. You might remember when we did this before, we want to fill our beaker up to 50, 160, 70, about 180 milliliters of distilled water. And then to that, we're going to add 20 milliliters of household bleach. So we're going to fill that up to the 200 mark. And then we want to give this a little swirl here. Notice I have my lab coat on again today, of course. I'm wearing my new button. I like not having polio, pro-vaccine button. And I'm going to just mix this a little bit with, I would use a pipette if I were you, but I had these little wooden stakes around. So I do need to disinfect my work surface. And really, something that we haven't talked about a lot, really the bleach solution, they're actually light sensitive. And so that's why bleach comes in an opaque bottle, right? Like this, like milk, because the bleach is actually sensitive to light. And so if after a couple of days, it's not very effective, not as effective. But looks like I was a little too liberal with my drizzling of bleach. So it's going to take me a couple paper towels to get this to be dry. So yesterday, you might remember, on day 3, we completed our staining labs. And I was just going to show you real fast how mine turned out. On my simple stain, it turned out great. You can see the purple when I stained with crystal violet and the red for the saffronin. And then I did take a look at those in our mini microscope, and I could see the bacteria and their shapes. However, my gram stains, they were crap. So these are my gram stains, and I'll hold them up against the white of my lab coat, and you can see there's no color to these stains, hardly at all, maybe a little bit of purple. So I'm going to repeat those with the second set of slides. Remember how we made 2 sets, set 1 and set 2? So I'm going to repeat that later today. And I think you guys should not feel bad at all if you look at your first set and you think, I think I'm going to do that over. My advice to you in the do-over, and I think I put it in one of the announcements, is that my advice would be no water. I would just use 2 or 3 loopfuls of bacteria and don't put a drop of water on there. You'll get a more concentrated amount of bacteria in a smaller space. All right, so today is mostly prep work for the next day for our last lab, the biochemical lab. I read through that this morning and I found out that in order to do that lab, biochemical tests, we need trypticase soy slants of all 3 of our bacteria. Not all of the 4 originals. We are not going to use Serratia anymore, but we do need 3 slants of E. coli, 1 of E. coli, 1 of Micrococcus luteus, and 1 of Staph epidermidis. I used 1 of my slants, and I know some of you may have had broken slants, so maybe you didn't have enough. I used 1 of mine repeating. See, I've got 2 sets of exercise 2 because I did 1 and forgot to hit the record button. So I had to repeat. So I'm 1 test tube short. So I do have extra nutrient agar plates. I still have 4 remaining. So even though I had 1 that was contaminated. So I did pull out a nutrient agar plate. And do you see how I divided up into thirds here? And I labeled each third 1 E. coli, 1 Micrococcus luteus, and 1 Staphylococcus epidermidis. And I'm going to, with an inoculating loop, I'm just going to streak some of my broth culture on there, as well as in these 2 tubes, I am going to put, because this is TSA, I'm going to put the Staph and the micrococcus in these tubes, E. coli will grow anywhere and on anything. The E. coli will grow great on the nutrient agar. The staph and the micrococcus luteus, maybe, maybe not. So I'm going to double those up. So I'm going to use my last 2 TSA slants for micrococcus luteus and the staph epidermidis. And then I am going to put E. coli with an inoculating loop. I'm just going to smear it all over that. And I'm just going to go through the demo of that really quickly with you today. And then tomorrow, which will be our day 6, if these cultures are grown up enough, then we can take a look at setting up the last lab. I also, when I did the streak plates for isolation, I did multiple plates. So this is the Serratia plate from the first experiment, and it's just starting to turn pink. The Serratia actually should be like a pinkish color, and the Micrococcus luteus should be yellow. So this is just starting to turn pink because it's kind of chilly up here in this room where I've been doing the lab prep. So I'm going to bring them to a place in my house that's a little bit warmer, and hopefully then tomorrow we'll be able to take a look at these and record our results. Same for the streaking for isolation. It actually did a pretty good job here. You can see my quadrant 1, 2, 3, and a little bit into quadrant 4. However, they're not pink and yellow yet either. So again, I'm just going to keep these aside. My broth cultures look good from aseptic technique. But again, the Serratia is not turning pink, so I am going to give it another day. I'm going to put it in a warmer place and give it another day. So I'm going to set those aside again. But the disadvantage is I'm still carrying around all these tubes in my test tube rack, starting to get kind of crowded in here, right? So I took my box, the TSA, the tryptase soy media box, and the lid, it came off. And so it had a perforation here. So you can use this like a test tube rack. So that's what I'm going to do. I'm going to place my micrococcus and staph epidermidis in here, and I'm going to move my tubes close to them. And then as I inoculate them, I'm going to do what I always do, move them to the back of the rack. I'm going to use this for my rack today. I did parcel out my E. coli, and I put my Serratia in a separate container so I don't even look at it and grab it by accident. I did parcel out my E. coli because I am going to put that on the Petri dish as well. So let's go ahead and get started here. Oh, darn it, I forgot my flame starter again. I'll have to pause the recording. My apologies. I have to pause this recording and run downstairs and get my... Okay, back with my lighter. So take my little cap off my paraffin burner again. This lasted longer than I thought, which was also a good thing. So you can probably leave it lit more than when I keep turning it on and off when we're doing our demos here. And again, you did not see me wash my hands before I began the lab, but that's of high importance to always wash your hands. And I'm wearing my lab coat, but I am going to turn back my sleeves on my sweatshirt here so that if I splash anything, it's going to splash on my skin or on my lab coat rather than on my clothing, because the bleach, of course, would ruin it. All right, so we're just going to continue practicing our aseptic technique. Because we're doing transfers today, I'm not wearing gloves. I prefer not to. It's totally up to you. You can go ahead and wear gloves, especially if you have cuts on your hands or your skin's super dry, which it's turning that time of the year. So I totally understand. But I prefer the tactile sensation of actually touching the things myself and just washing my hands very well when I'm finished. So this is the last inoculating loop in my first bin here. And I'm going to transfer the E. coli first onto the Petri dish because this is a one and done for the E. coli. For the other 2 bacteria, I'm going to inoculate tubes as well as the plate. So I'm going to do this one first. Remember, you want to be sure that before you do that, before you shake, you close the cap. And I think you can see the turbidity in the solution now. I should have brought up an empty tube that was uninoculated, but I did not. Remember, when we take the cap off, we want to turn the test tube, not the cap. So I can just hold my pinky still. I'm going to reach in here and grab a loop full. I forgot to flame. 2 points off. If I was doing it in the virtual lab, right? So flame the top there. Close this up. And I'm going to put my E. coli out of the way so I don't accidentally grab it. Now I remember I labeled on the bottom of my plate where the media is. I have my plate sitting on the lid of the dish. So I'm just going to pick up my dish and I'll come a little closer so you can see a little better. And I'm just going to lay E. coli in a zigzag wide at the top and get skinnier toward the center. I'm going to take this still full bacteria. Trying to see how well that doesn't tap very well out of these loops. So I just gave it a good tap. And now I'm going to pop this into my isopropanol to kill the bacteria, any bacteria that might be still on that loop. Next, I would label my test tubes. And so I'm going to be labeling them. This is TS broth. So this is Micrococcus. I'm going to label the tube Micrococcus. And I'm also going to put the date on here. So I believe today is the 14th. And it would be appropriate to put your initials on there as well. And I'm going to label the other tube Staph S epidermidis. And the date as well. Marker down, and now I'm going to have to transfer a tiny bit of my broth, just like we did in the aseptic technique lab. I'm going to transfer broth onto the slant, and I'll go over that again with you guys. So now I'm starting my second pack of loops, and it's a full pack. So again, I'm going to be sliding the loops down. I'm going to pinch and grab onto one of these loops that's way at the top here so that I retain it. And then I'm going to lay my package down, carefully open the package, and push that single loop that I had protruding toward the surface. Just going to kind of shake it. I used to just shake them out like this. Of course, they're not cooperating, so I'm going to hold down everyone else. Actually, I'm going to hold that one and send everyone else back down again. I can't get that loop. See, I got it just to come out all by itself. Grab that guy. Close my container to keep it as sterile as possible. And let's start with the Micrococcus. So I'm going to close the tube, give it a really good shake, because Micrococcus doesn't grow as well, as fast as the E. coli. So if you would actually even compare them to each other, with the E. coli on the left here, you can see how much more cloudy that is than the Micrococcus. So I want to be sure to give that Micrococcus a good shake. And then I'm going to remove the cap by twisting the tube. I'm going to flame this time. Insert my needle without my loop without touching the sides. And I'm probably going to put 2 loopfuls on just to be sure. So I'm going to close this, and I'm actually going to, this one says Micrococcus on it. Double check, Micrococcus, Micrococcus. I know it's backwards to you. Turn, flame the loop, flame the top of the tube rather. And then these are slants. So remember, we go in and we lay our loop down on the butt, to the butt of the tube, and then we draw a zigzag up to the top. And as I mentioned, I think I'm going to do this twice. Why, you ask? Because these bacteria, see how I'm holding the culture I'm starting with between my thumb and my forefinger? I just flamed that, so I doubt it really needs it again, but I'm going to grab a second loopful here, flame, and deposit it into this tube. Notice I closed this cap tightly on the broth tube that time because I don't want the broth to spill while I'm inoculating this. Give that guy a little flame at the top. Excellent. Close him off. And I'm hanging on to my loop here because I'm going to open that a quarter of a turn, put this in the back of my rack, but I still need my micrococcus because remember, I'm also going to lay it out on the Petri dish. So I'm conserving my loops. It's less time consuming if I don't have to go back in to the bag and try to fish out a second loop. So I'm just going to take a big loop full of bacteria. Make sure I have a loop full, flame the top there. Now I'm going to put the micrococcus back into, well, I'll put it back there to the back of the rack. And I'm going to, on the area that's labeled micrococcus, I am going to smack a little bit because I want that bacteria to come out of the loop. So I don't know if you guys can see, but I can get a little drip there. And then I'm going to spread that around the Petri dish, just in that quadrant. This has bacteria on it, so I'm going to pop it into the isopropanol. Now I've done the E. coli and the micrococcus on my Petri dish. So I'm going to turn my dish and I still have the staphylococcus. I'm just going to repeat the procedure that I just completed. And I'm going to try to do it all with a single loop. I find it's easier to grab onto one of the loops, and then when you open the package, I'm forcing it from the bottom outward, so I don't have to put my fingers into that little sleeve very often. And then actually at the end of the lab, which you guys haven't seen, I've taken the disinfectant on my paper towel and I've cleaned out that little slot in here where you can fit your finger in so that it doesn't get contaminated over the days that I'm using it. Okay, so here's my Staph epidermidis in my TSA broth. It's a little more vigorous than my micrococcus. It's going to show all three of these to you in sort of decreasing order. So the three that we're transferring today, E. coli, Staph, micrococcus, right? So Staph's in the middle here as far as how quickly it's growing. But I'm going to close the lid down and give it a little bit of a shake. It's best if you just kind of do that rocking, gentle rocking. Whoops. Use your pinky, Dr. Stewart. Twist the tube, flame the top. Again, I'm going to put it in the test tube first, and I'm probably going to, again, take 2 loopfuls. When you reinsert your test tubes into a rack, you don't want to let them drop because that'll send the bacteria up into the cap. You want to be sure that you set them completely down into the container, into the test tube. And I'm going to unscrew my TSA slant here that says Staph epidermidis on it because they labeled it correctly. And again, I'm going to try to smack a little bit because I want to get that bacteria out. Close this guy real quick. And actually, I'm going to keep a hold of him. Grab my Staph, my stock culture, flame the top of this. Grab a second loopful. Flame the top because I'm probably not going to be using this again. And I'm going to actually remove that and put it at the back of my rack. And then I'm going to concentrate on transferring. You can't lay these test tubes down on the surface of your lab bench. They have to go in and out of the rack. And you can't hand them to someone else. Like in the lab, a lot of times students want to hand it to their lab partner. And they say, oh, can you hold the tube while I do the cap? And no. All right, closed. And then I back it off a quarter turn. Put it also in the back of the rack because now the front of my rack's empty. My work's complete. I still need to put a drop onto my Petri dish though. So I'm gonna go back to my Staph epi tube, give it another little light shake, flame, grab a loopful, flame the tube, close the tube, move it to the back, pick up my Petri dish again, still laying on the lid, pick up my Petri dish, and try to do this so you guys can see it a little bit. I'm going to kind of tap the bacteria on there and then wiggle that drop around so that I get a good growth smear. This still full of bacteria. Pop that into the isopropanol. I'm finished with my flame. I'll turn that off. I don't want the isopropyl to catch on fire. And again, I'm going to take my original cultures, keep them back in my original rack here. My new subcultures, these are my subs, and I'm actually going to label them sub for subculture. And this is my subculture plate. And I should put the date on this so that I know when I started the cultures. I'm going to put these 2 test tubes and this Petri dish in a warmer spot. I'm not going to leave them here on the countertop like I've been doing. We'll put them someplace nice and toasty warm so that they'll grow up enough for us tomorrow so we can do the biochemical test. The biochemical test lab is our last lab. It's exercise 6. We're going to set it up on what will be day 5 then, and we'll hopefully can read the results day 6 or day 7. So even with the extra days, this whole procedure, not the microscope portion, but the whole procedure of actually executing the experiments will take us on average 7 to 9 days. So that's why I reiterated to you guys that there were 2 weeks before Thanksgiving to work through the labs, or 2 weeks after, or if you want to start them, it's anywhere in between. But just remember, every day, even Turkey Day and Friday after Turkey Day, you do have to check on your experiments and then set up the next materials for the next experiment. So once you get started, it's best if you work all the way through them day by day rather than waiting, especially on the gram stain. If the cultures are much older than 24 hours, you'll get incorrect staining colors. Gram positive cells will start to stain gram negative, and gram negative cells will start to stain gram positive. They'll give you false results. So it is kind of important that we do it all. The fresher the cultures are, the more likely the results we get will be clean and will be accurate. So I'll see you guys again tomorrow.